Hello, Mr. Allman here, and this is your Allmancast on taxonomy. Taxonomy is the science of classification. We need to put things into groups because there's a lot of life out there, and it's really hard to be able to understand all of that without being able to organize and sort and categorize all the living things that we come across. And once we have that level of organization, it's a lot easier to be able to attack important questions. Questions about where it is, when it is, that life came about. In this video, we're going to take a look at both old and new ways in which we have classified organisms over the years. And our modern classification system really has its roots in the 18th century with Carl Linnaeus. Linnaeus was the first to come up with a hierarchical classification system in which species were grouped together by shared morphology. So the more common characteristics that any two species shared with each other, the more likely they were to be placed into the same group. These system of nested groups are what we still use today. He also developed a common convention for how we should name living things. The system of binomial nomenclature utilizes two parts: the genus, with the example here of Homo, and the species name, which has sapiens. Together, the scientific name for humans would be Homo sapiens, with Homo being the genus, which would be capitalized, and the whole thing being underlined or italicized. Now, this gives some advantages. Not only does it allow for scientists in different places in the world who utilize different common names a way to be able to communicate accurately about the specific living thing that they are studying, but also because the species name includes the genus, that gives some important classification information embedded into its name. Because if you know the name of the species, then you can already identify other living things that would share some common characteristics with it. At the top, Linnaeus placed kingdom as the largest of the groups of life, and he identified 5 different kingdoms. The number of kingdoms, though, has changed over time, and we have actually supplanted kingdom with a larger group known as domain. There are 3 domains: bacteria, eukarya, and archaea, and it is mostly cell anatomy and an important molecular biology that actually distinguishes the living things in each of those 3 domains. Overall, we have just shy of 2 million species that have been described in scientific literature. The estimated total ranges anywhere from just shy of 10 million to up to 1 trillion living things. So even the most conservative estimates say that we have a long way to go in terms of identifying the forms of life that exist on Earth. And knowing that there are many millions or tens or hundreds of millions of living things still out there, having a flexible and robust classification system will certainly aid in our ability to be able to place new living things that are discovered into our existing framework. We can place those 3 domains onto a tree. This phylogenetic tree shows the bacteria, the archaea, and the eukarya, of which we are members, together as 3 branches on one single tree. The very first cell would not have been necessarily any one of these 3, but instead the first forms of life diverged through speciation into these 3 groups. You can notice that the archaea and the eukarya appear closer to each other on that tree. So now our modern classification system, in utilizing those domains, we have a bacteria domain, archaea, and the eukarya do still have kingdoms. There are 4 kingdoms which line up with those several of those original kingdoms that were in the earlier classification systems. Now, before we take a look at the new system of classification, it's helpful to understand why it is that we need a new way to classify living things. Well, there were problems with the old system. The number of kingdoms, the number of phyla, and the number of groups at every level of the hierarchy kept changing. They would increase over time as new species were being discovered. And it became clear that the debate over what constitutes a new group was inherently biased. Visual observations of living things is what led to these groups being formed in the first place. A decision to be able to choose characteristics that were important and then sort the species based on those characteristics. The problem is that looks can be deceiving. Two scientists can look at the same organism and come to different conclusions about which characteristics it has. The described species that we have are heavily macroscopic. We've done fairly well on describing the animal and plant species present on Earth, but the number of fungus and bacteria and protists lack far, far, far behind. And so we need a classification system that's capable of being able to not only sort things based on these large visual characteristics, but on cellular and these submicroscopic criteria. Just as an example of that, we could look at and we could see lots of shared characteristics between these two species of mole. However, if the characteristics that we choose are based on the presence of fur, the shape of the body, the type of limbs that it has, then we're going to group them together. But some of the behavioral and some of the anatomical differences that make this one a marsupial and this one a placental mammal, then we're going to see that these things actually belong in very different groups. And that gets us to a fundamental difference in how we classify. It's not just about choosing characteristics and then being able to sort species, but rather it's about looking back to evolution, to look to see where it is that these species came from in the first place. If we looked at how the hominid species are grouped based solely on morphology, when the characteristics are the things that lead us to make groups, you can see how the tree sorts out humans, chimps, gorillas, orangutans, and gibbons. However, when we use inference of evolutionary history, which uses a variety of evidence, we see a reclassification, and so the relatedness between species is still the same, but how it is that those species are organized within the tree is now very different. And this new tree is actually more helpful in being able to describe where it is and how it is that these species came about, because each branch shows a point at which one group separated from another part of that group. The revolution really comes with DNA, because DNA is a much more reliable indicator of relatedness. While characteristics may or may not be important features, and scientists can argue and debate about the merits of choosing one characteristic over another, when utilizing DNA, there is much less need for inference and interpretation. The DNA is a set of code, and that code is what it is. And that code can be compared from species to species. The term that used to describe the science of that is called phylogeny. Phylogeny is about determining evolutionary relationships, and that science of determining those relationships can then be applied to taxonomy to be able to sort things in a much more reliable natural classification system. While radioisotope dating with fossils gave a start in terms of answering the questions about when it is that certain things evolved, DNA has proven to be much more reliable and accurate in being able to provide that evidence. The reason why that we can look at DNA to be able to determine that is because the rate of mutation is fairly predictable, and we can look and we can chart and see the number of mutations in a particular gene, and then to be able to infer, based upon the number of mutations observed between two different species, we can infer how long ago it is that they actually evolved. So once DNA began to be sequenced and analyzed in this way, the field of cladistics was born. Cladistics is a way to classify things using that natural classification system based on shared derived characteristics from a common ancestor. The example of the pentadactyl limb is a shared derived characteristic, because the common ancestor of all land mammals had a pentadactyl limb and passed on that characteristic to its offspring. The more shared characteristics that species have, the closer to each other they're going to appear on a diagram called a cladogram. To make a cladogram, you start by looking and charting out how many species have a particular characteristic in common. Important characteristic for how it is that we classify land animals is shown here on the right. The presence of a backbone, of hinged jaws, of 4 legs, of amniotic eggs, and hair. If a particular organism has that, it has a 1. If it lacks that characteristic, it has a 0. Then those species are grouped together with the most characteristics in common. So when you look, you see the turtle has 4 out of the 5 listed characteristics. The leopard has 5 out of those 5 characteristics. Because the turtle and the leopard share the most number of characteristics, then they would appear next to each other on that cladogram. Additionally, the cladograms may include little symbols that are labeled to show the development of a new derived characteristic. If hinged jaws appears at this part of the tree, anything that branches off to the left of that hinged jaws mark doesn't have a jaw. But anything that appears to the right of it, past that hinged jaw mark, is going to have it. So the fish, the frog, the turtle, and the leopard would all have hinged jaws. So when you look at the tree, you can learn a lot about the organism, about how it evolved, and which characteristics were important markers in its evolution. 2 important mathematical principles need to be applied in order to interpret the DNA evidence. Because when you look at a particular gene or a genetic sequence and analyze it between 2 or more different species, you can get percent similarity that there is between those organisms. Maximum parsimony is the principle that we use in cladistics to be able to say that the simplest explanation is true. So shared characteristics are then assumed to be likely to evolve from a common ancestor. If there is a 30% difference between a human and a mushroom, and a 40% difference between a human and a tulip, because a tulip is more different from a human than the mushroom, then it makes more sense to put the human and the mushroom next to each other, and to say that 15% of those differences that exist between them in their genetic code, those are things that evolved separately after animals and fungus branched off from each other within the eukaryotic domain. When we look at cladograms, we can then define groups. The goal is to produce monophyletic groups, where we have a group, like group 1 here, that are all descendants of a common ancestor. Species A, species B, and species C are all closely related and would share a lot of common characteristics with each other, and so this is a sensible group for us to talk about. Group 2 is incomplete. This is a paraphyletic group. This group, some of the descendants came from a common ancestor, but we are leaving out some of the others. Other species or groups of species that also came from that same common ancestor. Group 3 is the most problematic. A polyphyletic group is one that contains species that descended from a diverse set of common ancestors. While A, B, and C all came from a recent common ancestor, species D did not, and therefore it doesn't make sense under the cladistics method to be able to include D as a part of A, B, and C. The classification of reptiles and birds can give us an example of how it is that cladistics can change our understanding of living things and where they came from. Traditionally, reptiles and birds would have been put in separate groups. They don't share many obvious characteristics. Reptiles have scales, birds have feathers. But deeper analysis, initially through the fossil record and backed up by studies of anatomy and of, more recently, DNA, have shown the many similarities that birds have with certain groups of reptiles. And this has led us to be able to see that reptiles is actually a paraphyletic group, because that traditional classification of birds in a separate group is incorrect, and that birds actually appear on the cladogram with those extinct and modern reptiles. And so if you think of a bird as being something that is related to a dinosaur and has a reptilian ancestor, that's going to change a little bit how you view the development of the different features of that bird, knowing that it came from a reptile ancestor. The new field of cladistics certainly has been problematic in that it's brought up many examples of how the older system of classification was flawed. And while it will be a time-consuming effort to be able to reclassify living things using this new system, the idea is that cladistics and the DNA evidence that it utilizes is going to be more robust and flexible, so that as new species emerge and are discovered and described, they're still going to be able to fit into the modern classification system without having to rewrite all the textbooks. So that was your Almond Cast on taxonomy. Please let me know if you have any questions. Thanks for watching.